Good morning, ladies and gentlemen. My name is Njabulo Gumede from University of Technology. Today's talk is entitled Augmented Intelligence Screening and In Vitro High-Throughput Screening Assessment of Drug-like Antagonists Targeting Androgen Receptors Allosteric Binding Sites for Advanced Prostate Cancer. Prostate cancer is the leading cause of cancer-related mortalities all over the world, including here in South Africa. The cancer registry report from 2010 up to 2019 has shown a marked increase in the number of men that have prostate cancer here in South Africa. So what happens is that most of these men are detected late at a stage whereby the cancer has already been at a stage where it has moved to other parts of the body. Like, for example, it has metastasized. In other countries, like the first world countries, the intrinsic resistance is caused by the mere fact that some of the compounds or novel antiandrogens grew resistance after 2 or 3 years of therapy. Therefore, this resistance is due to androgen receptor gene aberrations or androgen receptors plus variation, that for example ARV7 plus variant, where the ligand binding domain is diminished. So these gene aberrations causes missense mutations, which also renders the therapeutic ineffective. Like, for example, darolutamide has recently been approved for non-metastatic castration-resistant prostate cancer, and the overall survival rate also is also poor, because after 36 months it grew resistance to these particular interventions. So in most cases, there are 3 different types of cancers at advanced stage, like MHSPC, MCRPC, and NMSCRPC. Cancer stem cells drive these processes, like for example the survival as well as metastasis, which then is responsible for the resistance to conventional therapies. Therefore, this is an unmet clinical need. The discovery of compounds that will target these resistance mutations as well as the splice variations. So what we have done is that we have used an AtomNet approach in collaboration with some scientists from Atomwise, where we traverse the ultralight library of drug-like molecules, and then we targeted a groove region in the androgen receptor, where there is a presence of amino acid tyrosine 739. So this is an allosteric region whereby it is not susceptible to mutation as the ligand binding domain. So we traverse the chemical space by using a deep neural network, whereby we clustered the top scoring compounds, and then they were selected for synthesis. And then after that, after the compounds were synthesized, then we developed an assay whereby we use high throughput screening facilities with a robotics liquid handling, whereby in the master plate, the first thing that we did is to determine whether the liquid that is dispensed is of the amount of the compound that we intended. For example, for each well we have to add 45 microliters of the liquid. So the first thing that we did is to determine whether the liquid handling is able to dispense the amount that we intended for it to be dispensed, and then that process was passed, and then we then move over to a single dose concentration assay where we screen the compound. So the results are given here in figure E. So the cutoff was 40% growth inhibition, whereby all of the electric compounds were tested on healthy cells, right? And then the cutoff was below 40%. And then for hormone resistant cells, hormone sensitive wild type cell lines, as well as the androgen receptor mutations, we selected all the compounds that have a percentage growth inhibition that is below 40%. Now, to further explain this, we use a Venn diagram. In this Venn diagram, it shows that 8 compounds were sensitive to androgen receptor mutations. That they were selected, including the compounds that were sensitive to AR null inhibitors, as well as to wild type androgen receptor. Therefore, we ended up with 18 compounds, including the non-active compounds, as well as 2 lead compounds, as well as the 3 positive control inhibitors. So before we did the dose response experiment, we measured the overall assay performance of the secondary screening, whether it is robust or not. So we use the average Z-score approach, whereby we determine whether the DMSO, the 10% of DMSO that was used for the dilution, has an effect on the cells. Does it react with the cell, and to what extent? And then as a score of around 0.61, with a good correlation between replicates, is good for repeatability and reproducibility. So after that, we then treated the cells for 72 hours, and then we measured the cell growth inhibition using cell titer glue cell viability assessment assay. So we measured these compounds at a 7 dose concentration from 1.56 to 100 macromolar. And then we measured the IC50 half maximal concentration. So what is important here is that, as expected, the positive controls like darolutamide and enzalutamide were the cell lines, all the cell lines were resistant to them. And compound 1, 8, and 77 were the compounds that were selected because they had an IC50 value that ranges between 1.56 to 15 macromolar. So we will then further explore those compounds for mechanism of action, as well as PK and PD, as well as efficacy, using xenograph models. I'll explain that at a later stage. So these are the key findings that we managed to get out of this particular research. So we identified 3 compounds that are going to be further explored, as I've explained earlier on. So what next? We have to now do a lead optimization in silico using FAP plus in order to assess the relative binding affinity of the compounds, so that we optimize these compounds by doing R group enumeration. And then after that, we assess them via in vivo and in vitro efficacy studies and pharmacokinetics and pharmacodynamics studies. And then by 2024, we envisioned that we will have finished the study and done the IND registration. So these are the researchers that have helped in this particular work. So lastly, I would like to also highlight the fact that we wanted now to expand the patent estates of our design ideas so that we can be able to license our technology to other biotech companies. Thank you very much for your time.